a multidisciplinary study across 3 different lethal coronaviruses using a suite of technologies from proteomics to virology to clinical data. This is a collaboration involving 200 scientists from 14 institutions and companies in 6 countries. Not all are here, but we have representation from 12 scientists representing 5 academic institutions and 2 companies. Let me introduce you to the panelists in alphabetical order. Jared Carlson-Stevermer and Kevin Holden from Synthago, Adolfo Garcia Sastre from Mount Sinai, Robert Gross and Svenja Ulferts from University of Freiburg, Nevin Krogan, Oren Rosenberg, and Clement Verba from QBI UCSF, Andrew Pedden from the University of Sheffield, Jeremy Rassin from Aetion, and Veronica Rezel and Marco Vinuzzi from the Institut Pasteur. I will now hand this over to Nevin Krogan.
Okay, great. Can you see my slide?
Yes.
All right, thanks, Jacqueline. My name is Nevin Krogan. I'm a professor and director of the Quantitative Biosciences Institute at UCSF, and it's my pleasure to give you an update on some work in the context of the QCRG, the QBI Coronavirus Research Group. And as Jacqueline alluded to, there's a paper that was just recently published in Science about an hour ago. Comparative host coronavirus protein interaction networks reveal pan-viral disease mechanisms. And there's a number of authors, as you can see on this paper, almost 200 authors. And it really is, I would argue, a multidisciplinary worldwide collaboration. Even more impressive, I would argue, that it was done during the course of a pandemic. And just to briefly allude to 2 other papers that have come out of QCRG over the last few months, which are connected to this particular paper. The first one was published in Nature a couple of months ago, a SARS-CoV-2 protein interaction map revealing targets for drug repurposing. We generated a protein-protein interaction map, looked at the human proteins that physically connected to the viral proteins, and then made predictions about which ones could be drugged through drug repurposing. So this is a host-directed approach. And on the right, even more recent, there was a paper published in Cell. Again, we're looking at potential host-directed therapies where we globally looked in infected cells at a process called phosphorylation, which is carried out by a set of proteins called kinases that are very druggable. And this allowed us to identify a series of other drugs and compounds that we and others are looking at as potential antivirals for SARS-CoV-2. And a lot of the authors are the same in all 3 of these papers, and it's been a great honor to work with so many great scientists around the world. The red dots here correspond to the institutions and the companies that have participated in these works. So you can see it is truly an international effort. And here's a list of the partners, both academic and in the industry setting, that we have been working with over the last 6-7 months on these studies. And for me, one of the silver linings of this pandemic is the realization that when we all work together, we can move incredibly fast. And the challenge is to try to keep this infrastructure in place so that we can be better prepared for the next pandemic. So the highlights here of this particular paper, as Jacqueline had said, it's a comprehensive multidisciplinary analysis of all 3 lethal human coronaviruses, SARS-CoV-1, SARS-CoV-2, and MERS. And this allowed us to identify viral vulnerabilities and commonalities that could lead to antiviral drugs for SARS-CoV-2, but then also in the future for pan-viral coronaviral therapies. It's a worldwide effort involving 200 scientists and 14 public and private institutions in 6 different countries. We were the first to uncover the importance of one of the SARS-CoV-2 proteins that actually exists in SARS-CoV-1 as well, ORF9b, and its role in the inhibition of human immune response. We actually have a cryo-EM structure of ORF9b binding to a human protein, which I'll show you in just a few minutes. And we've also tried to do something unique in this study, connect the molecular data to the real world data using 740,000 medical records from COVID-2 infected patients. That's about 10% of the Americans that have been infected with SARS-CoV-2, so it's a massive data set. And we use this data set to confirm clinical impact of selected targets that we've identified. This was done in collaboration with Aetion, the company, and Jeremy Rassin from Aetion will be speaking about this in just a few minutes. And finally, we've identified several therapies and druggable targets that we and others will almost certainly be studying in the future. So just a quick overview of the study. We looked at SARS-CoV-2 previously, as I talked about, and in a similar way, we also then looked at SARS-CoV-1 and MERS with respect to generating a protein-protein interaction map. Each virus has between 25 and 30 proteins. And what we did with SARS-CoV-1 and MERS, just like we did with SARS-CoV-2, was to put a tag or a hook on each one of the proteins, introduce them into cells, pull out the tag or the hook on the protein, and then see what human proteins are physically connected to each one of the viral proteins using a process called mass spectrometry. This allows us to generate protein-protein interaction maps. In parallel, we took each one of these proteins, put them into cells, and using microscopy look to see in which organelle does each viral protein localize to, because if you know where something's residing, this can be very insightful with respect to its function. We merged this information together, and then we also carried out genetic analysis on the interacting proteins, including CRISPR-based genetic analysis, structural analysis, which I alluded to before, and I'll show you in a minute. And then, as I said, trying to connect the clinical data with the molecular data to identify certain trends that could be beneficial for future therapeutic strategies. So this was the SARS-CoV-2 human protein-protein interaction map that we had published a few months ago in Nature. The red diamonds correspond to SARS-CoV-2 proteins. The circles are human proteins. If the circles are orange, we think that they're druggable targets. And in this particular study, we identified 69 different drugs or compounds that we predicted could have antiviral effects through drug repurposing. And we evaluated their antiviral efficacy and toxicity in collaboration with scientists at the Pasteur Institute and at Mount Sinai in New York. And this allowed us to narrow in on a number of drugs which we're excited about. I just want to highlight a couple, including the translation inhibitors, pladepsin, aplidine, and zotadifin. So pladepsin, aplidine is from a company, PharmaMar. It was being used to treat multiple myeloma. They have a clinical trial ongoing right now for COVID-19. They're getting very interesting and exciting results, which will be released soon. And then also zotadifin. We just recently got approval from the FDA to start a clinical trial with this particular drug. It's from the company Effector. This is also being used to treat multiple myeloma, and it has very potent antiviral effects in the cell. And then we're also very excited about sigma receptor modulators. We have sigma-1 on this map, and I'll get to this and show you how it's conserved across different coronaviruses. And I just want to highlight that there was a piece in Nature Biotechnology saying that there are 13 drugs from this paper that are now being tested in clinical trials, and actually many more since that date. So this study, I would argue, has been very fruitful in that regard. So obviously, we're in the middle of this particular pandemic, the SARS-CoV-2 pandemic, but I think it's very valuable to look in the past in order to look to the future. And as we're all aware of, there's been 2 other lethal coronaviruses that have been problematic since the turn of the century, including SARS-CoV-1 and MERS. They have 8,000 and 2,500 cases worldwide, respectively, with a much higher death rate than you see for SARS-CoV-2. But obviously, there's been a lot more cases of SARS-CoV-2 because it's more transmissible, and a lot of the people are actually asymptomatic. So it's making it very hard to contain. But one of the logics here of this study is we wanted to look at the trajectory of the biology across these different viruses so that we'd be in a much better position for the next pandemic, for SARS-CoV-3, because it's coming. We know it's coming, and hopefully we can be more prepared for it in the future. So in that regard, just like we did with SARS-CoV-2, we generated a SARS-CoV-1 human protein-protein interaction map, highlighted here. 25 different SARS-CoV-1 proteins, identified over 350 human proteins. Similarly, we've generated a MERS map using the same approach. We got 22 of the MERS proteins to behave, identifying almost 300 human proteins. And there's a lot of comparative analysis that one can do, very exciting comparative analysis. This is just one simple way to do it, which is actually quite powerful. It's just to cluster the protein-protein interactions. Each blue line here corresponds to a different human protein. And the 3 groups that we're most interested in right now is this group here, interactions that you see across all 3 coronaviruses. That's about 1/5 of the interactions. This group as well, about a quarter of the interactions correspond to interactions you only see with SARS-1 and SARS-2 and not MERS. And then finally, 1/5 you see with MERS that you don't see with SARS-1 and SARS-2. And one of the ways you can look at these different maps is through this process called differential network mapping. Here's the math behind it, which I won't go through. But what we're doing is essentially looking at these three clusters, interactions you see in all three viruses, just with SARS-1 and SARS-2, and just with MERS. And there's a color scheme here corresponding to each one of the edges that corresponds to each one of these groups. So if the edge is black, it means it's an interaction that we see across all three coronaviruses. If it's blue, it's specific to MERS. And if it's red, it's specific to SARS-1 and SARS-2. And just to highlight a couple of clusters here that we've been looking into in greater depth. So one of the proteins here, NSP6, in these three coronaviruses, we find it interacting with sigma R1, the receptor that we've been focused on. It's very druggable, but we only see it with SARS-1 and SARS-2 and not MERS, hence the red edge. In comparison here to the coronavirus protein N, we find a number of black edges saying that these interactions exist for all three viruses, including a connection with casein kinase 2. This is a enzyme, a kinase that we'd identified in our previous two works. And this is important because there's a drug here, simultasertib, which is an FDA-approved drug to treat a cancer. There's now an ongoing clinical trial for this for COVID-19. Based on this map, we think that it would have effect not just on SARS-CoV-2, but SARS-CoV-1 and MERS as well. There's a number of connections here also with RNA processing, and therefore we think that these other drugs that I alluded to, plidepsin and apladine and sotaterfen, that are now being looked at with respect to SARS-CoV-2 could have effect on other coronaviruses, including SARS-CoV-3. And just one last connection here I want to highlight. This is a MERS-specific connection. This is involving the NSP14 protein. And interestingly, only in MERS do we see it interacting with proteins involved in DNA damage response and cell cycle arrest, including a number of cancer genes, including the most well-characterized cancer gene, P53. So it remains to be seen what is the reason behind this, but these are particular interactions that we and others will now be looking into in greater depth. Now, we've also, as I said, looked at the localization of each one of these viral proteins across these three viruses, because if you know where a protein is residing in a cell, this often gives you key insight into functional properties. And this is work spearheaded by Andrew Pedden in Sheffield, as well as Robert Gross and Svenja Elfritz in the University of Freiburg, as well as with Adolfo Garcia Sastre. So what we did was put in each protein one at a time just to see where in the cell it localized to. And then actually Massimo Palomierini in the University of Glasgow in Scotland, very excitingly, has been generating antibodies against SARS-CoV-2 proteins. And he's created 14 different ones against 14 different proteins. And we use those to localize the proteins in infected cells as well. And this is going to be a great resource for the community, these particular antibodies. So this work has led to a number of images and a lot of information, which will be fed to the scientific community today. And here's just a quick summary of the localization patterns as we look across the three different viruses and the different viral proteins. Here I'm showing you the localization score when you just introduce in the individual proteins. The more blue it is, the more it's localized to that particular compartment. You know, the cytoplasm, the ER, the Golgi, etc. And what I'm showing you here is the localization pattern based on the antibodies that were generated for each protein so that we could do the localization in infected cells. And I'm summarizing this here, strong, good, or weak, highlighting on this particular heat map, the blue heat map. There's some overlap, there's some differences, but there's a lot of information here and some great tools that will be very valuable. I want to highlight one connection here. It's with ORF9b, which I talked about before. You can see that in SARS-CoV-1 and SARS-CoV-2, it localizes to the mitochondria. And it's pretty much the only protein with these two viruses that localizes to the mitochondria. So I'll show you an image here. This is from Robert Gross's lab. This is in infected cells. We're using the antibody against ORF9b generated from the Palomierini lab in Scotland. And what I'm showing you here is essentially a quintessential localization to the mitochondria. And the mitochondria is the workhorse of the cell. It is the organelle that generates energy for the cell. So it's a very important organelle for the virus to be hijacking and rewiring during the course of infection. And we think that it uses this protein ORF9b in order to accomplish that. And I'm going to come back to ORF9b in just a minute when we look at the structural studies. All right, so in parallel, what we've also been doing here is genetic validation of the SARS-CoV-2 human protein-protein interaction map, looking at all 332 interacting proteins for SARS-CoV-2. This is work being done by Marco and Veronica at the Pasteur Institute, who are on this call as well. And so what they did was use an approach called RNAi interference and knock down each one of these genes one at a time in lung cells, A549 cells expressing ACE2. to see what effects they have on infection. And they used a readout based on the RNA component of the virus using RT-qPCR. Right? So that was one thing that we did. And then in parallel, we collaborated with the company Synthigo with Jared and Kevin, who are also on this call. This is a company in South San Francisco. What they did for us in a very generous way is use CRISPR to knock out each one of these genes one at a time in a different cell type, in CACO2 cells. Of course, Jennifer Doudna, our colleague here in the Bay Area, won the Nobel Prize for CRISPR last week. And Synthigo used this approach to delete each one of these genes. And in collaboration with Chris Bassler at Georgia State University, he then determined what effects the knockouts had on infection in this particular cell type. And the readout that he used was an antibody readout looking at an anti-N antibody. So in total here, we're looking at all 332 genes with 2 different cell systems, 2 different genetic perturbation strategies, 2 different assays, and 2 different continents to give us confidence that the data that we're getting is reliable. And here's a summary of that data. In total, we identified 73 genes out of the 332 that, when knocked out or knocked down, had an effect on infection. In red here are genes that had an effect in A549 cells. In yellow, the ones that had in CACO2 cell. In orange are the ones that had effect in both cell types. You can see sigma R1 here in the middle. This is a very good summary here, I would argue, of the genetic analysis, which identified 73 genes, about 20% of the protein-protein interaction map that had functional relevance in the context of infection. Many of these are druggable targets. And here's a summary of that particular data. So each blue node corresponds to one of the 73 genes. And then I'm showing you in this representation the connection to the corresponding coronavirus protein. So the SARS-CoV-2 here in red, SARS-CoV-1 in orange, and then MERS here in yellow. And I just want to highlight one particular connection right now. It's a connection between ORF9b, which I told you about before, and a physical connection with TOM70. I showed you before that ORF9b localizes to the mitochondria. Well, consistent with that, we find that ORF9b protein physically interacts with TOM70, a mitochondrial protein. So this is all consistent, suggesting that ORF9b and SARS-CoV-1 and SARS-CoV-2 is somehow manipulating the mitochondria. And there's been a group here, a subgroup within QCRG, the Structural Biology Consortium, which is led by Oren Rosenberg and Clem Verba. Again, we're also on this call. And they have consolidated, I would argue in a very unprecedented way, 60 different scientists, researchers here at UCSF, mostly students and postdoc. And they're working together incredibly collaboratively, generating these structures that normally take years. They're really doing it in a matter of weeks and months. So they're marching through these maps, structurally characterizing these connections, mostly now using cryo-EM. And they generated this particular structure, which I think is incredibly interesting. The green protein is TOM70, and the orange protein here, the orange structure corresponds to ORF9b. So you can see that ORF9b is tucking itself up into the active site, which is known as the active site of TOM70. So what is TOM70? It's a mitochondrial protein. It's involved in shuttling proteins in and out of the mitochondria. So we're still looking into why is ORF9b binding to this protein. Maybe it needs TOM70 to get itself into the mitochondria, or maybe it's manipulating other host proteins in a way that is beneficial for the virus. And if you take a closer look at the structure, there's some interesting trends. It's mostly hydrophobic, with 36 out of the 38 residues interacting directly with TOM70. And there's 2 residues here I just want to point out, 2 serines, S50 and S53, which we found actually in a previous study to actually be phosphorylated. That was in the Cell paper. And this to me is very interesting, because if this protein is phosphorylated here, there's no way that ORF9b is binding to the active site of TOM70. So we think that these phosphorylation sites are inhibiting or breaking up this interaction, which could be beneficial for fighting off infection. And we're looking at this in greater detail. I just want to show you one more video here of ORF9b, because I think it's incredibly rare if you look at its biochemical and biophysical and structural properties. When it's by itself, one of the molecules binds to the other one of the molecules, and so it's called a dimer, and it has a very distinct configuration. It's comprised mostly of beta sheets. This is what I'm showing you right here. However, when it binds to TOM70, it adopts a very different configuration. It's mostly alpha helices here configuration. So I think this is incredibly rare where you have one protein looking so different depending on the different context. And I think this is a major reason why SARS-CoV-2 is so problematic because its proteins are coming in and having very different shapes and thereby impinging on or affecting many different pathways and processes in our cell during the course of infection. So we're really not dealing with 29 proteins. I would argue we're probably dealing with a lot more. So one of the things we're doing right now in the QCRG is looking at this structure and making predictions about drugs and compounds that could come in here and bind and inhibit this particular connection, and hopefully that would have therapeutic value. And as I said, Clem and Oren, they have this great team and they're marching through these interactions and getting structures now very regularly that will be released from the QCRG in the near term. So the one other set of connections I want to highlight from this particular map here is the connection between sigma R1 and NSP6 from both SARS-CoV-1 and SARS-CoV-2. And also PTGSE2, we actually find it connected with NSP7 from all three coronaviruses. And both of these are important because they're both very druggable. And on the right there, we've actually shown a number of sigma R1 modulators having antiviral effects, including haloperidol, which we'll talk about in just a minute. And then also PTGSE2, which is prostaglandin synthase. It's involved in the prostaglandin pathway. And it's known that a pain medication, indomethacin, binds to and inhibits PTGSE2. So we made these molecular connections, and now this has allowed us to try to take this into real world clinical data in collaboration with Aetion. And I'm going to let Jeremy Rassin just finish up the presentation with this particular analysis.
Thanks so much, Nevin. So Jeremy Rassin, President and Chief Science Officer of Aetion, really glad to join the call this morning. Good morning to everybody. So as Nevin said, what we sought to do was to take those hypotheses and test those in real world data. So Aetion is a company based here in New York City. We focus on looking at the effects of medications in real world data. Real world data is the data that comes from the routine operation of the healthcare system. So as Nevin said back at the beginning, we created a data set with 740,000 COVID positive patients. And that data set included medical and pharmacy insurance claims, lab results. We can go back to the slides for a sec. Lab results, electronic medical records, and then this kind of data called hospital charge master data, which is a really detailed view into what goes on within the four walls of a hospital in terms of the services that patients are getting. So we created this de-identified data set. We identified treated patients and controls. And so looking at those two treatments that Nevin showed on the last slide, indomethacin, we identified patients newly treated with indomethacin in an outpatient setting, matched them to patients newly treated with celecoxib, another NSAID medication, but without the hypothesized COVID-19 benefit. And similarly for haloperidol and more generally the typical antipsychotics, matching them to patients newly treated with atypical antipsychotics. And so we matched two ways. We matched on time, and that's both calendar time, because there's been so much change in the way COVID is treated over the last 6 months, as well as disease progression time, because we wanted to make sure we were doing an apples to apples comparison where we looked at each patient starting at the same point in their disease progression. And then with each of these two groups of patients, the indomethacin group and the typical antipsychotic group, we were able to see whether, compared to their controls, the severity of COVID-19 was reduced among the treated patients. Next slide, please. So here's a schematic of the first of those two analyses, the indomethacin analysis. And working across the top here, you can see we started with 736,933 patients treated in an outpatient setting for COVID-19 with confirmed COVID-19, and identified 244 of them who began treatment with indomethacin. So a relatively small number. It's not a drug that's super frequently used, but we also found then 474 patients treated with the control medication celecoxib, and through that matching process came up with a group that was comparable one to the other. You could see that they were fairly comparable to start with. That's the overlap area that's pretty substantial between the blue and the red curves in that first density plot, and then perfectly overlapped after the matching process was completed. And these were small groups, about 100 patients in each group, and a small number of outcome endpoints. So the outcome endpoint here for these patients who initiated treatment in an outpatient setting was whether they required hospitalization. And among the indomethacin patients, 1 required hospitalization, but 3 required hospitalization among the users who began celecoxib. So that gives you an odds ratio of about 0.3, 0.4, somewhere between a 60 and 70% reduction in the need for hospitalization. But you can see that the confidence limits are very wide, and this certainly requires more study to see whether we can confirm this with additional data. On the next slide, you can see the same schematic, but for the antipsychotic analysis. Starting with 28,338 patients treated in an inpatient setting, 1,100 of whom, roughly speaking, began treatment with typical antipsychotics, about 1,500 of whom, again, roughly speaking, began treatment with atypical antipsychotics, going through that same matching process, coming up with a good comparison group. And we can see that the marker of severity here used in an inpatient setting was not going into the hospital because they're already in the hospital, but rather requiring mechanical ventilation once the patients were in the hospital. 13 of the typical antipsychotic users required mechanical ventilation as compared to 26 of the atypical antipsychotic users. And that gives you, again, an odds ratio about 0.4, 0.5, so roughly a 50% reduction. So again, early data with relatively small numbers, but an indication potentially of possibilities here, and also I think a real demonstration of how these hypotheses that come through all of the work that Nevin described can be assessed in a real-world setting with patients treated for this disease. So thanks very much.
Great, thanks, Jeremy. Just to emphasize too that we found antiviral effects with the typical antipsychotics in the laboratory setting, but not the atypical. And we think that typical work through sigma-1 and atypical don't, which is a great synergy with the clinical data that Jeremy just showed.
Yeah, thanks for the clarification. Similar to the indomethacin and celecoxib with hypothesized effect in them.
So just to summarize here, some key points. I described a multidisciplinary research that really is only possible, I would argue, with this global scientific collaboration. And I really want to see this type of spirit stay in place as we prepare for the next pandemic. We've identified shared vulnerabilities across the most recent lethal viruses, SARS-CoV-1, SARS-CoV-2, and MERS, and then elucidated potential for pan-therapeutic strategies. The data today is obviously relevant to SARS-CoV-2, but as we've been alluding to, this also may serve to expedite response and therapies to the next viral pandemic. And I just want to thank all the collaborators that we've been working with, the ones on this call and the ones that aren't, and the ones around the world as we augment this very exciting collaborative train. I'm very excited to be working on the next projects with this group, including not just the most senior scientists at these premier institutions, but the team of researchers who represent the next generation of scientists, the young scientists, and the future thought leaders. So thank you very much for your time.
All right, so I think we'll segue into the Q&A section. Please type your name, affiliation, and question in the Q&A box at the bottom of your screen, and we'll ask the panelists your questions. In the meantime, oops, sorry. In the meantime, before we get our first questions, perhaps I can ask one. How were you able to coordinate these efforts around the world during a pandemic?
Yeah, maybe I'll chime in and let others also after. I mean, to me, this is, as I was alluding to, this is one of the more remarkable aspects of this. And at least on our end, at the Quantitative Biosciences Institute, we had set up a number of these collaborations before the pandemic. You don't just pick up a phone and say, let's work together. So we had a number of these relationships in hand before with the Pasteur, with Mount Sinai. So it takes time. It takes trust to build up these relationships. So I would argue we're in a really good position to respond to this particular pandemic. And hopefully, as I said, we'll hopefully be better prepared for the next one as well because of all the relationships that we've strengthened over the last several months with this pandemic.
All right, thank you. We have a question that's come in from Carl Zimmer with the New York Times, perhaps guided to either Oren or Klim, but Nevin, you can guide. Can you speculate on how the virus might be manipulating mitochondria?
Sure. I don't know. Klim, do you want to chime in and give us our thoughts on that?
Sure. Yeah, I mean, so I want to preface that, it's still a very active area of our research right now, so we don't completely know. But in the past, 20 years, mitochondria has turned out to be found to be a central, innate response, innate immune response sort of organelle, where a lot of interferon response is stimulated at the surface of the mitochondria through a series of large protein complexes. And we know for a fact that this protein, TOM70, is involved in establishment of those complexes and interferon response. And so the simplest sort of model is that the virus goes and inhibits that interaction one way or another to stem the innate immune response, which we know from a lot of clinical data and other studies is SARS-CoV-2 is very good at as inhibiting the innate immune response, and it's very important for it to do so because it produces a lot of double-stranded RNA during its life cycle. And so that's sort of one hypothesis, but there's definitely very many, we think the whole effect is quite pleiotropic, and so there are many ways this can play out. And I know, Oren, if you want to add to that.
No, I think that was a great explanation. And I think, mitochondria are playing a central role in the innate immune response, and TOM70 is a key protein on the surface. And so inhibiting it or changing its function is going to be important.
All right, thank you. We have a question from Tina Say at Science News Magazine. These studies in COVID-19 patients are very small numbers and not randomized controlled trials. Do you think your results justify starting such a trial, and should it be an inpatient or outpatient basis?
That's probably for Jeremy.
Sure. I mean, I think certainly these results suggest doing more research, and whether that's a randomized trial or more research with real world data, I think that's an open question. I think we can determine causality or determine a causal effect between a drug and an endpoint in real world data. We can certainly do that in a randomized trial. In terms of whether that takes place in an inpatient or outpatient setting, I think it's really about the drug and really about how the drug is used and what the endpoint that the drug could particularly prevent would be. And so I think that's really kind of a question of the design of the analysis once the particular question, the particular endpoints, and the particular structure is set up. But I don't have a strong bias one way or the other.
Can I jump in? I just wanted to mention, and this is all anecdotal for the time being, but on our end in Paris, we were testing various compounds, some of which were falling in the antipsychotic drugs, etc. And we also have been contacted by 2 hospitals in the Paris area with psychiatric services. Both were preparing for the lockdown, getting ready to separate all of their services, their hospital, in 2. And then we're surprised to see so few of their inpatients and outpatients that were undergoing certain therapies were coming in sick. And so since then, they've contacted us, and we're working with them to look at the list of drugs and medication that they were on. So I believe I believe they were preparing a first publication or description of this. And if it interests you, I can put you in contact with them. But we found it interesting that what was being seen on the ground, we were seeing it ourselves. So it warrants further study.
Thank you. We have a question from Science News Magazine. Can you discuss your IL-17 results and how that compares with previous studies of immune reactions among people with severe COVID-19?
Sure, I could maybe chime in on that. So IL-17 receptor we found in our protein-protein interaction map, physically interacting with ORF8. This is actually a SARS-CoV-2 specific connection because we do not see it with ORF8 from SARS-CoV-1. Through our genetic studies, this was actually work done by Veronica and Marco, when you use RNAi interference in A549 cells expressing ACE2, when you knock down IL-17 receptor, you get a statistically significant decrease of infection. So that's very interesting. And through some analysis done in collaboration with Pedro Beltreo at EBI, EMBL, we were looking at genomic information, specifically SNPs in individuals and how it correlates with COVID-19 outcome. And there was a group of individuals with a certain number of SNPs and IL-17, a gene that results in overexpression of a soluble form of IL-17, and this correlated with worse prognosis with COVID-19. So that's consistent with this particular receptor playing an important role during the course of COVID-19. We think we're getting at the underlying molecular mechanisms. We think it's through ORF8. Actually, Klim and Oren are now starting to look at this structurally, trying to characterize this cryo-EM. And almost certainly now this is a biomarker for prognosis of COVID-19. The question is, can then we turn this connection into something of therapeutic value? There are antibodies against this particular receptor. So we're looking at those right now. So this is clearly a key pathway. And as I said, we think we understand what the underlying biology is, and hopefully this will allow us to make bigger strides both on diagnosis, prognosis, as well as therapy as well.
Thank you. So following the therapy question from Genome Web, could CRISPR be used as a therapeutic strategy?
Yeah, I don't know if Jared or Kevin wanted to chime in on this.
Yeah, thanks, Nevin. I can take it. So I guess, first of all, CRISPR was used in this study really to kind of, accelerate the research. So hopefully we can get to a therapeutic and a drug that will work very effectively. On the other side, if you wanted to think about using CRISPR directly, I guess, as a therapeutic in people, that's really been, I guess, some of the work around that already has been started using a different type of CRISPR, the Cas13, which is an RNA editing nuclease. And so there's some groups actually Synthigo's been working with, like Stanley Key's lab at Stanford University, where they've actually shown this can actually be effective, at least in cells, and now they're trying to move into animals as well. If you're interested, there's a paper in Cell that came out. It's called PAC-Man, which stands for Prophylactic Antiviral CRISPR in human cells. And so there is a potential that CRISPR could be used directly as a therapeutic using a Cas13-based strategy.
I believe Jeremy had a little more to say on a previous question. Jeremy?
Yeah, no, Tina, I was just thinking about your question a little bit more, and just taking a step back. How can we think about looking at the effects of drugs in data, be that randomized or real-world data? And I'll say that the study that we presented here, certainly based on a novel virus, but the techniques used for thinking about how to assess the effectiveness of medications, the safety of medications in real-world data are well established. And so thinking about how we could follow on as new data accrue, I think we can think about doing that in a real-world setting like we did, or certainly in a randomized setting as well. It's not an either or. Certainly that these are both places where one could further evaluate the potential for these therapies in treating and reducing the severity of COVID-19. I'll also say, and maybe some of the other panelists can jump in here as well, that there are other medications that are either available or contemplated that may meet the structural criteria that have been described. And for those medications, we can also think about that same set of strategies for looking at the effectiveness in COVID patients. Again, a real-world strategy for drugs that are on the market, or a randomized strategy for drugs that are emerging. And so really, we could be pretty pragmatic in our choices here. And really, the idea is to get the information as quickly as we can, because this is certainly urgent, exigent, and important to understand.
And it's important to keep in mind that for these drugs, of course, the first goal is to repurpose what's out there to find something very quick. But also very likely, what this can lead to is that ultimately these repurposed drugs were developed for another thing. And medicinal chemists could work on those drugs to find derivatives or versions of it that are even better at coronavirus down the road. Of course, this would require more in vivo work to prove that that derivative also functions. But in this case, these kinds of studies have an immediate possible potential and a more downstream potential.
Thank you. We have another question from Genome Web, which I believe could be for Adolfo. You mentioned preparing for the future. Did the data tell you anything about the potential for patients who've had COVID possibly becoming reinfected at some point? And were you able to learn anything about the potential for SARS-CoV-2 becoming endemic in the population?
Yeah, it's not clear right now how prevalent are reinfections. They obviously have been documented, but whether they are very common doesn't seem to be the case. One of the cases that has been put in the literature has a person that after reinfection have more severe disease, which again it sounds like an unusual thing. Most of the evidence will indicate that the majority of the people that have already been immune due to natural infection are more likely not to get infected based on the proportion of infections that they are now in this proportion of people. And in some studies that I have done also in closed settings where a lot of people have been infected, but not those that have immunity. Having said that, however, it's very unlikely that the virus will disappear. And even after a good effective vaccine, which probably we will have at one moment, it's very unlikely that the virus will disappear because the vaccine is not gonna be 100% effective in limiting infection. And that means that the virus is gonna continue circulating even after having a lot of people acquire natural immunity or a lot of people being vaccinated with an effective vaccine. It's not going to cause the problems that it's causing right now with respect to the very rapid increase of severe infections that can happen because of the percentage of people that are still completely susceptible. But that means that it's going to continue, and then it's going to continue causing problems also now in a lower proportion of people. And therefore, I think it's extremely important to continue working on therapeutics, because the virus is clear that it's going to stay, not causing, hopefully, the problems that it's causing right now, but certainly still causing people some percentage of severe disease. So therapeutics will be great to have as in the weapons, even after a vaccine is already available.
Thank you. A question from BioWorld. Can you talk a bit more on the relative merits and drawbacks of virus versus host targeting?
I love hearing Adolfo speak, so I'm going to let him comment on that, and I'll let Marco speak after that on that topic as well.
So can you repeat, Jacqueline? I didn't get it.
Can you talk a bit more on the relative merits and drawbacks of virus versus host targeting?
Okay, yes. So that's quite important also. In the past, most of the therapeutics have been going to targeting virus, viral proteins, and we have very good success with this approach. You just need to see, for example, hepatitis C, or you need to see HIV, that we don't have a cure, but we have a way how to control it, and this is mainly with antivirals that target viral proteins. However, they have some drawbacks, the antivirals that target viral proteins. One is that they are extremely specific. So therefore, they are very good for a specific virus, but they don't have, in general, broad spectrum. And the broad spectrum antivirals that they are not very effective, not as effective as this one. Broad spectrum is, for example, interferon, right? That will be also an antiviral that targets the host by induction of an antiviral activity. It has some effects, but it has also limitations because of just the side effects that interferon induce. But the host has becoming more interested in the later on as an area of research. First, because it's now becoming evident with the science that we have that this is a valid targets, host targets that can be used for inhibiting viruses. And they have the potential to be broadly useful because viruses tend to use similar pathways and therefore similar host factors. And then they could be used in the case of potential new pandemic viruses that we don't know of at this moment. And also because in general, targeting a host factor makes more difficult for a virus to become resistant. Because the resistant for when it's targeting the viral protein is by point mutation in the viral protein, which viruses are very good at doing due to the high mutational rate. But targeting a host factor, the host factor cannot mutate. So the virus needs to mutate to overcome the need for this host factor. And in many cases, that's quite difficult for a virus to do so. So it's a very interesting area of research, but mainly I think because of the potential broadly applicability that inhibitors of host factors may have, again, multiple viruses in multiple families.
Marco, do you have something to add?
If I can just add that, indeed, the antiviral targeting of virus can be a magic bullet, but these are RNA viruses that mutate very fast, and at most they code for only a dozen or so proteins. So you only have a dozen targets. As we show in our work in this collaboration, we have hundreds of host proteins that interact, and therefore many more targets that we could use to drug. While the virus is a fast and violent intruder, the host is quite strong and resilient. And so for a lot of these host targeting therapies, what happens is these viruses have just a few hours in which to replicate and do what they need to do. We can hit a host protein, inhibit a key host protein for a certain amount of time and not affect the host cell. The host cell will be able to recover while the virus is in that 12-hour period missing key essential resources. So it's a little bit a game where we throw the advantage on the host that can win the long game rather than do the quick run that viruses try to do.
Thank you. And a bit of a follow-up question. Could you speculate on how many approved drugs are out there that could potentially modulate the disease course of COVID-19 based on the host factor patterns you've identified?
Marco, do you want to continue on with that train of thought?
No, I don't know exactly how many. That's for you to answer.
Well, it's a great question. You know, there's several thousand FDA-approved drugs. It's a great question. We don't know. I mean, there's been efforts where they've been screening them, all of them one at a time. Our approach is different. We have been creating a data-driven approach. We're trying to get at the underlying biology behind the SARS-CoV-2 infection and let the data point us in certain directions. They could be FDA-approved drugs. They could be drugs in clinical trials. They could be preclinical molecules. So that augments the search space for a drug and a compound even more. But I think our approach is one that will be beneficial in the long run, because the more you know about the biology, the more you could be effective in the pharmacological space. So more work's needed. We've got to get more biological information collected. It's these great collaborations that allow you to collect such valuable information to allow you to point you in certain therapeutic directions. But I'm hopeful that this group and groups like this will narrow in on some drugs that'll have therapeutic value in the near future.
What can be said is, I mean, it is difficult to speculate because we've never seen this before. So much of the planet trying to find any kind of antiviral. I have to say, on a personal level, I was surprised to find this many with our collaboration, right? So So I find it very encouraging, and it's who knows how many more are there in those libraries and banks that people are working through. Also, I think what is encouraging is that I think many of these antivirals that we find have some effect on these coronas are going to have an effect on other viruses, not only of the coronavirus family. So the spillover effect will benefit throughout virology.
Thank you. We have a question perhaps for Veronica. You used lung cell lines for your studies. Have you done something similar with immune cells to better understand how the virus interacts with the immune system?
Yeah, so no, we haven't. The short answer is that no, we haven't done this on immune cells. So A549 cells are immune, so they have a competent interferon response. The reason why we're using the cell line is in the very beginning of the pandemic, it was really hard to find relevant cell lines that we could work with. So initially in our first paper in Nature, we're working only on VROE6 cells. So this is a cell that is derived from monkeys. So it's not very relevant, but it's also a cell line that scientists or biologists use very commonly to study virus replication and to that's like generate virus stocks and so on. So it's a very commonly used cell line. So we, in collaboration with Olivier Schwartz's group here in Pasteur, so they made a cell line that's so this A549 cell line, that it's a lung cell. It's very commonly used as well. However, it was not susceptible to infection. However, when they overexpressed ACE2, the receptor for A549s, we could use that as a cell line to study virus replication and target using different drugs to assess which ones have antiviral activity. Of course, it would be ideal and great to work with immune cells, but also when we're working in such a high throughput manner, it's also very difficult. Ideally, you would want to use human primary cells, like lung primary cells and so on. But for such large studies, it's a difficult task. However, for some of the targets that we have, it would be certainly interesting to pursue this, especially with IL-17, for instance, that has where the immune response has a very important effect. So yeah, it's definitely something to look into in the future.
Maybe I'll just jump in. The cell type specificity, I think, is a very important one as you collect data. There's been some beautiful localization data in this paper in a couple of different cell types. Maybe I'll just call upon Svenja and Robert to discuss, as you go across different cell types, how would you envision the localization changing? From either Svenja or Robert. Oh, I think you're muted. Svenja, I think you're muted.
Hi.
Yeah, there you go.
Okay. So Robert had to leave. He had to go pick up his son. That's why I'm jumping in now.
Okay, great.
I wouldn't really expect a lot of difference, like with respect to localization of the proteins in different cell types. What we used are these cargo cells, which is a colon cell line, which we used for many practical reasons in the beginning. When we started imaging, these were the cell lines that we had at hand that were able to be infected with the coronavirus, because not all of the cell lines that we had were actually able to be infected because they were missing the ACE2 receptor. And they're actually very beautiful to image. So that's also very nice when you do like high resolution imaging, that you just have cells that don't grow on top of each other, and that are just where you can actually see the single organelles. We just started also imaging with a different cell line. We also used the Vero cells that were mentioned a few moments ago. And we don't see much difference so far with respect to localization. So I don't think that's a big issue.
Thank you. So we have just a bit of time left, perhaps enough for one or two questions. From Science News Magazine, the paper mentions that sigma-1 protein is also a target of antihistamine. Can you talk about what those antihistamines are, and if you see any clinical data to suggest those drugs might be useful as a prevention or treatment for COVID-19?
Yeah, that's a great question. Clemenstein was one antihistamine that we did show, that Marco and Adolfo's labs showed that they did have antiviral effects. I know, Jeremy, that you did, we did discuss looking at this in your cohort, but I think I think too many people take this, and I don't think you can get a properly controlled analysis of this. Do you want to say a few words on that?
Yeah, I mean, there are a couple things that make this particular study of antihistamines a little bit more challenging than some of the other ones. One is that many of these drugs are available over the counter, and so they wouldn't show up in pharmacy data. That's not an insurmountable challenge, but just in the time that we were working under, wanted to really prioritize the medications that were readily studyable in this population. The other is, again, like Nevin said, the broad use would require some kind of sorting out of patients to focus on very specific groups, and also a relevant comparator would need to be chosen, like we did with celecoxib for indomethacin, or the atypical antipsychotics for the typical antipsychotics. But again, none of these challenges insurmountable in any way, just prioritized some of the other medications given the urgency.
All right, thank you, Jeremy. So we've come to time. We have about 30 seconds left. Thank you, everyone, for participating, and I'll hand the mic over to Nevin for a few last words.
Yeah, I'd like to thank all my collaborators for being on this call from around the world. It's great to see all of you. Hopefully we can get together in person for some celebration drinks. I picked Paris, Marco, for you to host us. And also thanks to all the media that came on and asked such great, riveting, fantastic questions. So thanks everybody for participating in this, and I hope to see you all soon.
Thank you. And this video will be made live on YouTube at the QBI TV UCSF channel.